So now let's look at the output of those aligner. The most widely used format is called the SAM format. So in the SAM format, it will begin with some nice describe the reference genome and some commands you use to do the aligning and some other informations. Like for example, this some important is like the start with the SQ. It will tell you the chromosomes in your reference genome and the length of that. And each read will has one entry in the SAM format, and it will look like this. So there are some important columns in the SAM format. So the first column is the read ID, and the second column is some text, which tells you whether this read is aligned or not, whether this read has mate pair, has the other mate pair, and whether this read is aligned in a reverse complement way or in the forward way. And the column after that tells you the chromosome this read aligned to, and the coordinate of this read aligned to. And the next column is the mapping quality, which are defined different 2 by 2. So I don't think there's a consensus on the score now. And the next one is a very important field. It's called cigar string. It tells you how the read is aligned to the reference genome. Like this will report whether there's some indels in your alignment. And if there's an insertion, it will tell like, and if the insertion size is 3, this might be looked at. And the M here means it's a match or mismatch. And if there's an insertion in the middle of that, this string might look like 13. This might look like, oops. Yeah. And that means there is one insertion in the. Can we see that? Some other. Yeah, nevertheless, there are some specialized character to tell you there's an insertion or deletion there. And the most important for RNA-seq, which you probably will deal with later, the introns in the alignment will be represented by nucleotide by character N here. So when you look at the SAM file, you will see the intron like 1000 N. That means this read spanning 1000 base pair as intron. And then the column after that is how is mate pair aligned. So in this case, it's an example of single end reads, so they are not held there. And then this is the nucleotides for that read and its quality score. And each tool will have some auxiliary field in the BAM tool, so you probably need to check they usually start with character X or Y, so you probably need to check their manuscript yourself. And remember that in SIGA string, it only tells you this base pair match or mismatch. It just means they are on the same coordinate. So this MD field in the column tells you what's the difference if there's a mismatch. And for this one, it will say there are 3 base pair match, and then there's a mismatch of C to the reference genome. And then there are 30 base pair match, and then there's a T mismatch. in the reads is T, but in the reference is other. And though you can, if you, and this field are very useful if you are doing like SNP analysis, like the variant analysis, and because you can see the read is different from the reference genome. And then this field start with NM is, you can remember that a number of mismatch, which says this read has 2 mismatch to the reference genome. And some of them also has some other important fields like the NH, number of hits, that says this read is aligned to how many positions on the reference genome. Since there are some repeats on the reference genome, a read can be mapped to multiple positions. And you can use the second column, the flag, to tell whether this alignment, this entry is the same in the same file is the primary alignment for that read or not. And for the second read in the entry here, we can see since this flag is 4, it says this read is not aligned on the reference genome. And for this one, the NM is 0, that means this read is perfectly aligned. And all this information are in, there's a very, the SAM tools has a very nice README file, so you can check those yourself. And there's another format called bed tools, which are not usually not used often in represent the alignment. It is most used to represent the intervals, like to represent the genes on the human reference genome. Like says this part to this part is exon, this part to that part is some genes on the reference genome. And its format is very straightforward. It just tells you this interval is on this chromosome, and it starts on this position and on this position, some README for that interval, and others like score. And then some strand, whether it's the gene is on the plus strand on the reference genome or it's on the minus strand. And the bed tools, and for bed format, there's a very useful tool called bed tools. It can manipulate those intervals very efficiently. And sometimes you can use that to solve fairly complicated problems. So you can look into that tools yourself, especially that's useful when you're dealing with RNA-seq data. So here is a summary. So there are now currently 3 generations of sequencing technology, and we are most interested in the, and also in this course, we are most interested in the 2nd generation sequencing. And for those sequencing generation sequencing technology, Illumina is the most dominant one, so we will use their data. And the output from Illumina sequencing and also the other sequencing data is in FASTQ format. It has four lines, which tells you the sequence, the nucleotides of that read and its quality. And anytime when you have the data set, before you do any downstream analysis, it's always a good idea to run FastQC to see whether this data set is good or not. If it's bad, it does not. It does not worth your time on analyze that bad data set. And then we talk about some very technical stuff, how we retrieve the coordinate of the reads in the from the reference genome, there are 3 general strategies. One is use seed, where we directly store the coordinate of each chimer in the reference genome, and the suffix array and Boris-Wheeler transformation are all based on the idea of suffix. And the suffix array are using one of the most popular tools, STAR, in RNA-seq alignment. And the Boris-Wheeler transform are used in BWA and bowtie. And all the aligner, most of the aligner will generate the output in SAM format. And if you also compress that, it's called a BAM format, which are usually much smaller than SAM format. And you can copy that to many places. And I want to thank Liwei and Benamid and Eduardo for the slides they provide. And that's it.